Hello, and a very warm welcome to everyone joining us for today's Select Science webinar titled Improving Quality of Bacterial Culture and Oligonucleotides with UV-Vis Spectroscopy to Accelerate mRNA Vaccine Production. My name is Finn Price, and I will be moderating today's presentation. I'm delighted to be joined today by Naimish Sardesai from Thermo Scientific. Throughout the webinar, please feel free to ask any questions you may have for him in the Q&A session at the end. You can submit your questions using the chat icon to the left of your screen. Without further delay, let's begin today's presentation.
So, hello everyone. Thank you for joining this presentation. I am the product marketing specialist at Thermo Fisher, focused on core UV instruments that include Spectronics, Genesys, and Evolution product lines. And today I'll be talking about how you can improve the quality of bacterial culture and oligonucleotides with UV spectroscopy to accelerate the mRNA vaccine production. So, as the COVID-19 gained steam worldwide last spring, few companies emerged as vaccine makers. They earned an FDA emergency authorization in late 2020. And then there was a push to make these vaccines at a record speed to supply them to masses around the globe. And one of the critical concerns raised regarding these vaccine manufacturing was the availability of the raw materials. So manufacturing RNA is a very complex process, and it requires several highly purified ingredients. Now their quality would affect mRNA turnover, as well as the vaccine production, as you can see in this schematic. And one of the highly purified ingredients is the DNA plasmid that are being isolated through the cell culture process. And it is important to grow enough bacteria to isolate adequate amount of plasmid DNA. So UV-Vis spectroscopy is a common technique used here to measure bacterial cultures at OD 600. And the other ingredient is the oligonucleotide, which are actually the building blocks of nucleic acid that are used by the RNA polymerase to synthesize desired mRNA strands. And spectrophotometers can measure oligonucleotides and their yield in the residual proteins at 260-280 nanometers, or you can say 260-280 ratio, and evaluate the quality of oligonucleotides during the final QA/QC step. So the agenda of this presentation is in front of you. So first, I will go over the two applications. The first application I would talk about is monitoring the health of bacterial culture using UV-Vis instruments and how to standardize the data across labs with your organization or within your organization. The second application revolves around quantifying the nucleic acids and determining their purity in the protein preparations. And finally, I'll be going over some benefits of the UV spectroscopy in the pharma space. So let me talk about the application where the UV spectrometers are being used in bacterial culture. So you can see the schematic, and here the bacteria can grow widely, or you can say widely different in different media. And monitoring this bacterial growth is one of the critical steps. So this figure shows a workflow of how UV-Vis Evolution or Genesys spectrometers can monitor the health of bacteria needed for vaccine production. So measuring the cell growth over time at 600 OD is a common practice. And that is because at this wavelength, the cells are not being altered, while the UV light has shown to cause mutations in bacteria, altering or destroying the genes of interest. So here at the bottom, we have listed the three common applications where the OD 600 measurements are being used. So when the cells are being inoculated in a fresh culture medium, they basically initially go to the to something called as a lag phase. And during this lag phase, the bacteria would adjust to the surroundings and the nutritional options in the media, and the cells increase enzyme production and they will grow slowly. Then they would enter a log phase where they begin to grow, and you can see an increase in the absorbance at the OD600. So they begin to grow rapidly and divide at a constant pace before entering the stationary phase. Now, these instruments, the UV-Vis instruments, can help you determine the stage of the growth of bacterial culture. In general, the cells should be harvested towards the end of the log phase, and in this process, the OD600 values of the samples are being measured to determine when this point has been reached on the growth curve. So these measurements will surely help you to ensure that the cells are being harvested at an optimum point that corresponds to an appropriate density of live cells. So we all know that the OD600 is a common technique used to measure the optical density of bacterial culture. But then the question arises, like, why do we have these challenges? So first of all, it is important to determine the optical density performance and the dynamic range of your spectrometer. And this is because you need to cover the entire growth cycle of the culture. So here is an example where we actually measured the OD values of the diluted and the undiluted samples with the Evolution 260 Bio. And these OD measurements were done every 30 minutes for around 9.5 hours. Now, you see the blue lines that represent the diluted culture, whereas green lines are the undiluted samples. And you can see that the diluted samples fall within the dynamic range of the optical system. So it's a nice linear, I cannot say the linear, but it's nearly linear than the undiluted ones. So if the OD readings fall outside the dynamic range of the instrument, first of all, it is important to dilute the cell suspension. So the second one is most of the organizations would try to replicate similar results in different labs with another spectrophotometer, or a lab may decide to replace an old one. Therefore, we need to standardize these measurements, making them reproducible across the organization. Now, why do different instruments give different results? So for turbidity samples, such as like cell culture, the OD600 value results from the light scattering rather than the light absorption. Therefore, the instruments with different optical setup will give different OD readings for the same turbid sample. And this can make the standardization protocol hard to follow. And therefore, if the results from different spectrophotometers are to be compared, they must be normalized first. And I can show you an example which I have here. So we compared the corrected OD values for each instrument and the cell counts. The OD600 values from the diluted solutions were multiplied by the dilution factor to get the corrected OD values before we plotted them in this data or in this curve. And if you see this plot, the instruments with similar optical systems produce similar OD curves over time, but with some difference. And that difference is because not all the instruments are alike. They are nearly similar in the optical systems which they have. So when we go further, we normalize this OD data by correlating the OD readings of the same sample on two different instruments. For example, I normalized OD data for the Evolution 260 Bio using the Spectronic 200. So we calculated the average of the OD ratios between the two instruments at each time point and used that value as a conversion factor. So with this method, we calculated the conversion factor for Evolution 260 Bio to be around 1.22. So you see the blue line, which is the uncorrected one. And if you multiply each point there by 1.22, it actually gives you the green line. So that is the normalized data between the two instruments which we get. But you have to remember that this factor is specific to a bacteria or species because the scattering effect depends on the size, the shape of the bacteria, and will also affect the conversion factor. So basically, I've gone through like how you can use the diluted samples to reach the dynamic range, and also how we can standardize different instruments so that you can use them across the labs or across the organizations. And here, what I'm trying to say is like Genesys spectrometers, they have many advantages. So Genesys instruments are first smaller in size, that is around 14 by 14 inches, and can fit in the cell culture hood. Plus, these instruments have an onboard software with a 7-inch color tiltable touchscreen. They have a high-definition display and an inbuilt printer. So after you get the results, you can directly print them in the hood. You do not have to attach these instruments to a PC or an external printer, making your job and that makes your job easy. So Genesys 180 has a double beam geometry with a reference cell, and it also comes with a cell changer. These software they control the accessories like Peltier, zipper, and probe, and the Vision Light software, which is connected with this instruments, they actually meet the 21 CFR compliance. So with this, I can say that that Genesys or the evolution instruments can measure the OD 600 values and determine the stage of growth of a bacterial culture and ensure that the cells have been harvested at an optimum point with an appropriate cell density. Then how to overcome the challenges, we talked about it. So it's important to dilute the cell suspension if the reading is outside the dynamic range of the instrument. Secondly, you can also normalize data between the two instruments that would help you replicate similar results in different labs across your organization. And finally, there are a lot of advantages of Genesys instruments. They meet 21 CFR compliance and are fit for the pharma applications. Now let's move on to the other application, that how UV spectrometers can determine the purity of DNA and RNA. Just to introduce this, normally the nucleic acids are quantified by measuring absorbance at 260 nanometers, while 260-280 ratio determines the purity of nucleic acids. Now, protein contaminations can overestimate the nucleic acid concentration by artificially inflating the absorbance at 260 nanometers, affecting the nucleic acid purity. Now, the proteins that are co-purified with the nucleic acid also absorbs light in the same region, that is UV region. And traditionally, we accept the purity ratios to fall between 1.8 to 2.2, as shown in the table. And these contaminations not only mislead the researchers by inflating the nucleic acid concentration, but also affect the downstream and the qPCR reactions. So protein as a contaminant in nucleic acid preparation contributes to absorbance at 260 nanometers, and this inflates the nucleic acid concentration. There are two kinds of ratios, 260 is to 280 and 260 is to 230. 260-280 ratio is widely accepted way to detect protein contamination, while 260-230 ratio can also be an indicator. Now, the challenge of using 260-230 to detect protein contamination is that many other common contaminants, such as buffers, salts, and polysaccharides, can also affect this ratio. Now, evolution instruments have been used in the final QC step to evaluate the yield of nucleic acid in protein preparations by measuring the 260-280 and 260-230 ratios, which I will be showing you in the following slides. So how the degree of contamination affects the protein ratios and quantification results. So we show that with the experiments here. So in total, we took 7 samples that were being prepared, in which 1 sample was a pure DNA, which is the red line at the bottom. You can see 3 peaks here, the protein peak at 280 nanometers, the oligonucleotide peak at 260, and then the 230 nanometer, which we assigned to the peptide backbone. Now, these other 6 samples which you see are the DNA protein mixture with varied proportion of protein concentration. And we used evolution 201 spectrophotometers for the study. And you could see that as you keep on increasing the protein concentration in the mixture, then it inflates the oligonucleotide concentration at 260 nanometers. So as the protein contamination increases, the nucleic acid concentration is also inflated. And you also see an increase at the 230 nanometers. Now, we can assign the peak of 230 nanometers to peptide backbone because we are using a pure protein here, and we do not have any contaminants. And this is how we actually conclude that the 260-230 nanometer ratio can also be used as an indicator for protein contamination. As the level of protein contamination increases, the nucleic acid concentration is inflated at 260 nanometers, and we saw that in the last slide. But the large amount of protein concentration is normally required before we can see a significant change in 260-280 purity ratios, and this table shows it. So the samples with nearly 69% protein by weight still has a purity ratio on a lower limit, but still an acceptable for use in downstream experiments. That is around 1.67. And as the protein contamination increases from 69% to 90%, this ratio drops from 1.67 to 1.20. On the other hand, the 260-230 purity ratio decreases, which might be another indicator, as we saw in the last slide, for the protein contamination. But in real scenario, this would actually contain the buffers and the salts and polysaccharides. So it would also contain other contaminants. Therefore, this ratio alone cannot confirm the protein contaminations. At the end, I'm going to share with you that how our instruments fit the pharma space. So EVO UV platform consists of 4 different instruments with increasing level of performance and features. Now, we used Evolution 201 for this study, which has a fixed wavelength, but we also have the Evolution 220, 260 bio, and Evolution 350 with variable bandwidth, and increasing levels of performance as well. These instruments are very reliable QC/QA instruments with a flexible software feature and a consistent performance as well. They also have a xenon flash lamp, so that many years of measurement without having to change lamps, that actually decreases the cost over years, unlike some of the instruments that use deuterium lamps with shorter lifetimes and are subjected to often changing lamps that can increase the cost and hazards as well. These instruments have a double beam, which means you can have a reference sample in the reference position for your measurements. That increases the stability of your results. These instruments use a software called Insight, which is a modern software and has a lot of functionality. The instrument meets the 21 CFR Part 11 and the pharmacopoeia compliances, which make them ideal for a good manufacturing practice QA/QC environment requirements. So going further, Evolution 350 has additional flexibility to your lab needs. Evolution 350 can be purchased with an installed mercury lamp. So normally pharma customers will use the mercury lamp to measure the wavelength accuracy of their instruments without needing to purchase and maintain the liquid standards, which need periodic calibration. And larger the sample compartment makes these instruments compatible with a wide range of accessories which are not available for the 200 series, giving it much more flexibility. At the end, I would like to say that the Evolution series spectrometers, they determine the 260-280 and 260-230 ratio to detect the protein contamination in DNA or RNA samples. They meet the 21 CFR and the pharmacopoeia compliance, and this is why they can be used here for the protein contamination studies. Also, they have an option of various accessories and also an option of pre-installed mercury lamp to eliminate any risk of compromised standards. So with this, I will conclude my presentation and I'll open questions. Thank you.
Just before we jump into our question and answer session, I'd just like to take a second to remind you all in the audience that it's not too late to ask any questions if you've still got them to ask. So the first question we have for you today is, what is the acceptable range for A260 absorbance?
First of all, I'd like to thank everybody for attending this conference. And to answer this question, I would say that you need to dilute your samples so that the reading is between 0.1 and 2.5. Otherwise, you are using the instrument in a non-linear range, and you may not even get accurate results. So if you want a proper measurement then of how much the DNA you have in your sample, then that would be the approach.
How do you determine CFU for a given OD 600?
So CFEs actually stand for colony forming units. It's the number of live bacteria, and therefore you would do a viable cell count plate of the bacteria, and then determine the number of live bacteria per milliliter of the solution, and then you can plot them as it was shown in the presentation against the absorbance.
Can I detect DNA contamination from RNA samples or DNA fragmentation from the original DNA using a UV-Vis spectrophotometer?
What I could say that although we have done this successfully with our micro UV-Vis spectrometers, we haven't done this yet on the Evolution series spectrometers.
Can we change the default data save location? Can customers still save data in different locations?
Yes, the answer is yes. Basically, we are using an Insight software which has a security administration function. So you can configure that path in the security or administration, and that can be saved as an auto backup file. So basically, the security leads to the 21 CFR Part 11 that actually complies with that, and that's why you have to choose that path to save the data in a different locations if the customers want to.
Can this technique be used to assess vaccine purity as part of release test?
Sorry, I missed that question.
Sorry, I'll just repeat that. Can this technique be used to assess vaccine purity as part release test?
So basically, the release, I imagine that there would be certain impurities like genomic DNA, proteins, and some unwanted isoforms. And what I can say that we have demonstrated how we can find the protein contamination from the RNA or DNA samples. However, we are still to actually see if we can do that with the genomic DNA or unwanted isoforms with the original DNA sample.
I've just had a couple more questions come in. I'll give you just 2 seconds to have a quick look at them before I ask any. So on that, unfortunately, there's no more questions that we can answer right at this very moment. But thanks again for today's informative presentation, and thanks to everyone for joining us online. Oh, sorry, no, that's one more question. Sorry for jumping the gun there. So the last question we've got for now: if you don't have a reference spectrophotometer to calculate the conversion factor for your bacteria species, is there another alternative to normalized data? Are there turbidity standards to do this?
So what I can say here is you have to have a reference spectrometer to actually correlate the results, because if you do not have it, then you don't need to normalize any data, because the normalization of data is the main idea is to have the same results across the organization in different labs. So if you do not have any other instrument, then basically you don't need to normalize the data.
Now, on that note, we'll wrap up the presentation here. Thanks once again for the informative presentation and for answering our questions. If you have any more questions in the audience, please feel free to email me at editor@selectscience.net, and I will follow up with these personally. Remember, you can also download a certificate of attendance from the Documents tab to the left of your screen if you need these. If you'd like to listen to today's webinar again, or invite a friend to listen, it'll be available to watch on demand in a few days' time. Goodbye, and thanks once again for joining us.